This conference will now be recorded.
Good evening, students. I'm Dr. Divya. So in our biology class, we were learning about principles of inheritance and variation, right? This is coming under genetics. So genetics is the branch of biology which deals with the rules of genetical inheritance and genetical variations. Okay. In genetical way, what and all characters are getting inherited or what and all variations are happening, that about all those variations and inheritance we are going to learn under genetics, right? So now we are learning about the principles of inheritance and variation. What do you mean by that? Principles of inheritance and variation. There are some laws which are governing the inheritance and variation of characters. So in this chapter, under this chapter, we are going to learn about those principles or the rules of that inheritance and variation, right? That's the way the progeny resembling the parents in morphological and physiological features. Okay, so such things, how a progeny resembles the parents. That's what we are going to learn under this, and the pattern of inheritance, everything we are learning in this. So, in the last, we learned about the terms like genes or factors. First, the genes were called as factors. Okay, then we learned about alleles. What are alleles? Different forms of same gene. The same one gene will be expressing in different forms. The different forms are called as allele. Okay. Then we learned about Mendel's experiment. What he did, Mendel's experiment, Mendel, he's called as father of genetics. And he started this genetical experiments. He started learning about the genetics of one experiment. And he selected Pisum sativa. Pisum sativa, it is the garden pea, that plant garden pea. The scientific name of garden pea is Pisum sativum. And Mendel selected this Pisum sativum as a study material. Why he selected that study material? Because of the contrasting characters and short duration of that crop. So if a plant material or the study material, if you are selecting, you have to think about the characters, the contrasting characters, because then only it will be easy for the study of the characters in further generations, right? If it is not expressed in a proper way, it is very difficult to find out the characters in the coming generations. So what we need to do by selecting a study material, what and all character, or what is the reason for this selection of that study material by Mendel? Why he selected garden pea? Because it was having several contrasting characters which we can study very easily in the future generations. Okay, so that contrasting characters, then based on one more reason. What was the reason? The short duration and the easy cultivation of that crop. If a crop was very difficult to cultivate, then for cultivating the future generations, it will be very difficult, right? So the experiment won't be successful. That's why Mendel selected a crop which can be cultivated very easily. Then another reason was the short crop duration. If crop duration is less, then we can generate 2 to 3 generations per year, so that the study will go very easily and they can study within 5 to 6 years. They can study the characters in the further generations. After crossing the further generations, they can raise, and in each generation they can study the characters very easily. That's how a study material should be selected. So these are the reason why Mendel selected Poisson sativum as the genetic study material. Then he developed several generations by selecting these 7 contrasting characters. For each and every character, Mendel selected 7 contrasting characters for the study of this genetical inheritance and variation. Okay, for each character. Then for selecting each character, he made separate crosses for each character. He selected 7 contrasting characters. He made separate crosses for each character. Then he developed several generations after selecting these 7 contrasting characters. Then he made observations and he did monohybrid cross by selecting 1 pair of contrasting character each time. Okay. Monohybrid cross means in that cross we'll be selecting single character for study. Okay. Monohybrid, mono, that word is related to single. The single character we are going to select under monohybrid cross. So Mendel did monohybrid cross for each character and he raised separate generations, separate crosses for each 7 character. Okay. So in a monohybrid cross, they'll be selecting single character. So that's how Mendel raised generations for different contrasting characters. Okay. And here in a monohybrid cross, what you can see in a monohybrid cross, Mendel selected plant height. Here with an example I'll explain. Plant height Mendel selected as a contrasting character. Okay. Mendel selected plant height as a trait or a character for his studies. And he crossed. Here you can see he crossed this tall plant and a dwarf plant, right? He crossed homozygous tall and homozygous dwarf, right? This parental line was pure line. This is the parental generation. In parental generation, these were pure lines, right? That produces gametes, capital T and small t. This will produce only one type of gamete. That's why Mendel selected pure lines. Pure lines means how many generations we are leaving for selfing or self-crossing also, it will produce only one type of gamete. These are called as pure lines. Okay, so he crossed homozygous tall with homozygous dwarf. So this parental generation was pure line. That was the first criteria for selection. Pure lines he selected. Then these pure lines produce gametes. See here you can see capital T as a gamete. Dwarf produced small t gamete, and that formed the combination. Here you can see these both gametes combined from the tall one gamete and from the dwarf another gamete. These combine to form a capital T small t combination. Okay, this is the F1 generation. Which one is the F1 generation? This here, this capital T small t. This forms the F1, first filial 1 generation. Okay, so in these combinations, all these produced tall. Okay, so in F1 generation, in F1, all showed capital T, small t combination. See here, only one type of gamete. So all will be having the similar combination, right? So this will be the genotype of F1. And phenotypic, what was the phenotypic condition? Or phenotypically, all these were tall. Okay, all these were tall. These expressed tall character. Okay, then from this, what he observed in the F1 generation, either one parental character is expressed. Here, parent 1 is tall and parent 2 is dwarf. But after producing gametes, when it got combined, in F1 generation, they got only one parental character. Phenotypic expression was only one parental character, right? And that produced tall plants. Okay. Then he allowed for self-pollination. Here it was self, mating self, because this heterozygous condition, right? This he allowed for selfing. He didn't do any cross-pollination here. It allowed only for selfing. Then raised F2 generation. See how he made F2? Here, this F1, he allowed for self-pollination and that produced two types of gametes, capital T, small t, capital T, small t, right? It's selfing. So same types of gametes from both parents, right? Two different gametes will be produced from this parent also, from this parent and from this parent, because it is selfing, right? Same pollination, same pollen grains are falling on the stigmatic surface. Then raised F2 generation. Here you can see the F2 generation. See, this is the F2 generation what he produced, right? So in F2, both the parental characters were expressed in F2. Here in F2, you can see tall, 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 and dwarf due to different combinations. Here, the capital T and small t combined to form. Here in F2, I'll show you here F2 generation. F2 means it's selfing. Selfing will express in this way. Okay, this is the symbolization of selfing. Then it will produce, that means what? Capital T, small t is crossing with the same thing. Okay, so this will produce two types of gametes, this parent, and this parent also will produce two types of gametes, right? So in a checkerboard, how we will write, if you are making a Punnett square, how it will be? One parent produce these two gametes. You can write here capital T, these are gametes. Usually we will express gametes like this. Then this is another parent, right? So here you can see different combinations. The first combination is capital T, this gametes, first two gametes, capital T and capital T, right? So both are dominant alleles. So how it will be? It will be dominant. So this will express tall plant, right? Next is capital T and small t combination, these combinations. So how it will come? Capital T, and if the dominant allele is present, we'll write that dominant allele first and second comes the recessive allele. So here also the presence of dominant allele is there. Definitely this also will be tall. Third combination again in the similar way, capital T, small t. So this also, here also the presence of dominant allele is there. So this also will be in tall condition only. And the last one, small t, right? Here two recessive alleles are coming. So the expression will be the recessive character, right? So here in F2, the genotypic ratio was one homozygous tall, then two heterozygous tall, then one homozygous dwarf, right? This is the genotypic ratio. Next, we will see the phenotypic ratio. Phenotypic ratio means how the character is expressed. This is the genetic condition. Homozygous tall. This will be this homozygous capital T, that will be tall. Then heterozygous condition. This also, while expressing, this also will form the tall character only, right? So here 1 plus 2, 1 plus these two, both will form, all these three shows the tall character, right? So here 3 tall, and 1 dwarf. Right. So the genotypic ratio is 1:2:1. Then phenotypic ratio 1. Okay. With the help of a Punnett square, how we can express a monohybrid cross and what is the genotype or F1. F1 genotype you need to learn. You should not have any confusion in this in F1 genotype. Then what about F2? All these combinations you need to learn, and the genotypic ratio and the phenotypic ratio, you should be clear about this. Okay. Then for all these monohybrid crosses, which representing all 7 pairs of contrasting characters, and Mendel got same result. Here we just took one character, right? For an example, we selected one character, plant height, as a character. But Mendel conducted for all 7 contrasting characters, he conducted the experiment in the same way. Okay. And he got the same result. For all the experiments, he got genotypic ratio 1:2:1 and phenotypic ratio as 3:1. For all the experiments, he got the same result. All 7 contrasting characters. Okay. So based on these experiments and with the support of this large data, he proposed some outputs of these experiments as law of inheritance, and based on that, he got all these results, right? So he made a common outcome or a common result out of all these 7 experiments. What he made laws of inheritance. Okay. So laws of inheritance, Mendel made after, or Mendel proposed the laws of inheritance as a result of this experiments. Next, with the help of this Punnett square, we can explain this Mendel's experiment. And what is Punnett square? Punnett square is a diagrammatic representation, or it is a schematic representation of a cross. For what we are using this? To calculate the probability or the probable, all possible or probable chances of a genotype. Okay. All probable chances of a genotype of a genetic cross that we can get from this. Without any confusion, we can easily get the all possible combinations of a genetic cross. Okay. So here, by using the same trait, here again we are using the same trait, plant height. Plant height as a trait of study. So here with the same trait we are using, by using the same trait we are going to explain this. Okay. So here in this you can see Mendel selected 2 contrasting characters. Okay, 2 contrasting characters or 2 contrasting traits. Plant height was the character he selected, and what was it? It was in 2 forms. There were tall plants and dwarf plants, right? Tall and dwarf condition for one trait. So these tall and dwarf, these were pure lines. Mendel selected pure lines for his experiment. Okay, that pure lines formed gametes. Okay, and these gametes here you can see that form gametes, similar type of gametes, capital T and here small t. Okay, the dominant allele here and the recessive allele here. Okay, and these gametes fuse to form the new combination in F1 generation. Here you can see the gametes. These are from tall and these 2 are from dwarf. Okay, and these form new combination in F1 generation. Here you can see the possible combinations. Capital T, small t, capital T, small t, capital T, small t, and here also the capital T, small t. But in F1 generation, all were tall, we said. Why it is tall? Because in all these combinations, there is a presence of dominant allele. Here it is there, here also it's there, in this one also it's there, and the 4th one also it's there. So there is a presence of dominant allele in all combinations. That's why it came tall. Okay, F1 generation, all plants were tall. Okay. Then this F1 generation, he allowed for selfing, right? Mendel allowed in F2. Here we are going to discuss about F2. So here in F1, all were tall. But here, when F2 comes, here you can see in F2, Mendel allowed this F2 plants for selfing, right? So from these, the possible gametes you can see here. Here in this diagram, you can see the possible gametes after selfing. Which and all the gametes here, capital T, small t, then capital T, small t. You can see all the possible genotypes in this. Okay, after the combination of these different gametes, you can see different combinations, genotypic combinations. Here in first, in the Punnett square representation of monohybrid cross, you can see the F2 combinations by which is made by Mendel. So this Punnett square you can see capital T small t here and capital T here capital T small t and here small t. These are four possible combinations of these gametes in F2, right? This was the experiment done by Mendel. So with the help of this Punnett square, we are representing that experiment. So in F2 generation, tall plant produced tall. This is heterozygous tall. That is the difference. This is heterozygous. This is not homozygous. In F2, it is in heterozygous condition. So the heterozygous plant produced two types of gametes, capital T, small t, and here again the heterozygous plant produced capital T, small t. So these are the four different combinations made by these gametes, right? So here you're giving this diagrammatic representation, right? With explanation also. So if we are giving this diagrammatic representation or schematic representation without explanation also, it will be very clear. So that is the importance of Punnett square. Okay, F1 and F2 without any explanation, it's very clear. Here we started the experiment with tall and dwarf plant. Then the possible gametes from the pure line we can see and different combinations of F1 generation. Then when giving for selfing in F2, you can see the possible gametes in F2 and all different combinations. Here you can see the phenotypic ratio, tall and dark. It comes 3:1. Okay, and genotypic ratio is 1:2:1. So from this Punnett square, it is very clear that without any explanation, we can make sure that possible gametes, possible combinations, and the ratio of genotypic and phenotypic ratio with clarity. So that's the importance of Punnett square. So based on all these experiments, Mendel proposed law of dominance and law of segregation, right? We told that based on all these F1 and F2 in all 7 contrasting characters, he proposed law of dominance and law of segregation. These two laws he proposed based on his 7 contrasting character experiments. Okay. Then, till now we were learning about the examples which are following this Mendelian rules or Mendel's law of dominance and law of segregation, right? So we were following all this Mendel's law of inheritance or law of dominance and law of segregation, how one character is dominating and how it is getting segregated. So, till now we were learning about all these things. So all of you know that in biology for everything you can see exceptional cases, right? So that's the speciality of biology. That's why biology is called a science of exception, right? Like usually we can say that stem is above soil, right? Stem we can see above soil. But when we are thinking about potato or corn, what is the contrasting thing in that? We can say that or we are saying that potato and corn, they are the underground stem modifications. This potato and corn both are stem modifications, modified stem, we used to say, right? But usually stem is above the ground or above the soil, right? So when this case comes, stem, these are called as stem only, but they are underground stem modifications, right? In another case, all living organisms have nucleus, right? All living organism cells have nucleus, right? But mature RBC, when it is getting mature RBC of mammals, that doesn't have nucleus. Okay, so that's why, so like this, you can find out so many exceptional cases in biology. That's why biology is called as the science of exceptions. Okay, there will be special cases. Okay, so it becomes an interesting area to study, all these exceptional cases in biology. So that becomes an area of research. Okay, so now we can see the coronavirus, right? So all this coronavirus, there are different mutants are forming. Exceptions are coming. If you are finding out one solution for that, there is another exceptional case coming. So biology is a case of exceptional studies. Okay, so that becomes another research interest. Okay, so let's come back to our point. So we are talking about the exceptional cases of Mendel's inheritance. Okay, Mendel's law of inheritance or the deviations from that law of inheritance. So when experiments were continued for different traits in different ways, they found some like F1 individuals that had a phenotype or F1 expressed the character which are not like both the parents, right? In Mendel's process, what we said usually in Mendel's process, it was expressing F1. F1 character was parental character, right? In the case of parental traits, any one parental trait was expressed in F1 according to Mendel's law of inheritance. So now we are discussing about the deviations or the differences or the exceptional cases from Mendelian principle or exceptions from Mendelian principle. Okay, we are going to learn the deviations from that. Okay, so experiments continued and in different way they conducted the experiments. So some F1 individuals in some experiments, some F1 individuals, they showed a special phenotype or expressed another character which are not like both the parents. Okay, there were some different characters in the F1 individuals which are not resembling the parental characters. Okay, those F1s were showing a blended trait or a mixed up character, like they were expressing in between character of both the parents. Okay, there were, see, by here I'll say a parent is tall. With the same example, we'll say a tall and a dwarf plant. In this cross, so after some experiments, what kind of result they got? They got a medium-sized plant. Okay, so this kind of expression. Okay, I'm not saying, just for an example, I said if they are getting a medium-sized plant, that is not according to Mendel's law, right? So this kind of expression, that is called as in between or a blended expression, that is neither showing the tall character nor showing the dwarf character. Okay, it is expressing an in between phenotype. The expression is different. Got it? So when experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of two parents. Okay, that was one in between expression in between the two parents. Okay, so the inheritance of flower color in dog flower, which is that snapdragon, that is the example to understand the incomplete dominance. So we are going to learn about incomplete dominance. Incomplete. See, from this term, incomplete dominance, from this term itself, it's clear that it is not complete. Okay. Here, incomplete dominance, we said, it is the best example for the incomplete dominance here. Exceptions of Mendelian principle. First, we are going to learn about incomplete dominance. And here, the example we are going to study is dog flower. Dog flower, it is snapdragon. Okay, it is otherwise called a snapdragon. So this is a good example for dominance. That dog flower or dragon shows incomplete dominance in flower color. Okay, in this flower color, in that character, it shows incomplete. See, in the picture here you can see parental generation, red color, true breeding red color, red flower was crossed with true breeding white flower. Okay, these are pure lines. These both parental generations are pure lines. Red color is crossed with white color flower, both were pure lines. Okay, so in F1 generation, here in F1 offspring that showed genotype of capital R small r, that means heterozygous condition. That is an heterozygous condition. Okay. So in F1 here, the gametes produced, this red color pure line produced capital R gamete, and the white, as usual, this also produced the smaller gamete, which is common. This is the normal type of gametes. When in F1 generation, when these both combined and it formed pink color flower from this heterozygous combination of capital R, small r, according to Mendel's law, which one we will expect? Which color we will expect according to Mendel's law? The red color, because here dominant allele is present, right? So according to Mendel's law, which color that should express? The red color. Red color flower should get produced. Okay. But here, just like the previous experiment, the dominant allele did not express the complete character. If according to Mendel's law, that should express the red color, right? Here red dominant allele is present. So according to that, that should express the dominant character. But here in this experiment, that didn't express the dominant character or dominant allele expression, that didn't give. Instead of that, that gave pink color flower. Okay, even though this allele, it was dominant, that did not express completely. Okay, over the recessive allele. Here dominant allele and recessive allele both are present. Even though dominant allele is present, that did not express that dominant character completely. Right. So there is an intermediate character or intermediate phenotype by the F1. So the F1 produced flower, but the character, the phenotype of the flower was pink. All were pink. This was an intermediate expression. Okay, so phenotypic expression was produced by the F1 instead of parental characters. Just now I said these are deviations or exceptions from Mendelian principle. So according to Mendelian principle, red color flower should be produced. But here, these are the exceptional cases, right? So here, instead of expressing the dominant allele, the F1, all F1 generation, all plants of F1 generation show pink color flower. That means they showed an intermediate character. Then the F1, when it is selfed, here you can see. Selfing means how it will be? Here saying the heterozygous that will undergo selfing means it will produce capital R, small r, capital R, small r, right? Then the plants of F2 generation, here you can see how the plants will be. F2 generation plants. Capital R, small r, capital R, small r, right? Here, first combination is capital R, capital R. Fourth combination is small r, right? Combinations and all will be same. All F2 generation, here three types. Here this is homozygous red, right? Capital R, both alleles are same. So it is in homozygous condition. So that is homozygous red. Here, then think about this one. This is homozygous. Here both alleles are same, smaller. So this is in homozygous white condition. Okay, because here you see the small r that forms the white color flower. Capital R forms the red color flower. So here this is homozygous red. Both alleles are similar. These are homozygous white. White, right? But when these two are concerned about these two, what you can say here, capital R and small r are they both are there, but these gave in between expression, right? This gave this here dominant allele did not express completely. There was an incomplete expression of this dominant allele, right? It is in heterozygous condition, so this formed pink color. Right, this showed the pink color in between expression. So they were in 1:2:1. Here you can see 1:1, capital R, then 2 capital R, small r, and 1 small r. It was in this condition, this ratio only. Genotype 1:1. Also, genotype was 1:2:1. Okay. So in heterozygous condition, that is this capital or smaller. Here, this combination is coming, right? Here, mixing or blending of both colors, red and white. This red and white, these are the parental traits, right? So this resulted in the development of a pink color flower, right? So here, phenotypic ratio. How you will write the phenotypic ratio here? Here, phenotypic ratio also. Here, phenotypic ratio also similar only. 1 red, 2 pink, and 1 white. Here the phenotypic ratio also 1 red, 2 pink, and 1 white. Correct? So here genotype same phenotypic ratio also 1:2:1. Okay. So look at genotypic ratio, 1 homozygous red. Here you can see one homozygous red. Here, the big ratio. Homozygous, then homozygous white. So both ratios feel
Divya, voice is not audible, Divya. Hello? Hello?
okay. So
Divya, unable to hear you.
here you got
Divya,
one capital R and one smaller gamete, and in the F1 generation, all pink color flowers for dominant and complete dominance. It was complete dominance. Am I audible now? Can you hear?
Yes, ma'am.
Okay. So that showed a incomplete dominance. When that allowed for selfing that produced two types of gamete, because heterozygous definitely it will produce two types of gametes. So capital R small r and capital R small r. And these are the possible combinations of F2 generation. Capital R, definitely it will be red color because both is, and here it comes in the heterozygous condition or both alleles are different, then the dominant allele is not able to express completely. They shows an incomplete expression or incomplete dominance. Okay, so here phenotypic ratio we will get one red and two pink. In heterozygous condition, when the red and white crossed, it is showing an in-between character or a blended character. In between color it is showing pink flower. In heterozygous condition, it is not showing complete dominant character. Of incomplete dominance it is showing. It that as a result of that incomplete dominance that shows pink flower or that forms pink flower. So here two pink flower, then last the homozygous recessive that forms the white flower. So here phenotypic ratio is 1:2:1. In the same time you can check the genotypic ratio. In homozygous red 1, then heterozygous. In heterozygous again 2, capital R small r, and in homozygous recessive condition 1. So here in this case, in the case of incomplete dominance, both the phenotypic and genotypic ratios is to 1. For your board exam, you will get these incomplete dominance and all the ratios you have to note down. You'll get so many questions from this. Okay. So you should not get confused. In do not complete dominance in incomplete dominance. Both phenotypic and genotypic ratios are same. You experiment represent. Okay. Next is dominance. Okay. So actually, what is this dominance? So let me tell you the concept of dominance first. What is the reason behind this dominant allele expression, or what is that dominant allele? What do you mean by the dominant allele and the recessive allele? So to answer this question, first we should know, or we should have an idea about what a gene does, or what is the role of a gene. Okay, so you know that all genes are having different informations. They have these informations for the expression of a particular trait or a particular character. So genes are expressing, or these are the reason for the expression of particular trait or character. Okay, so a gene determines the skin color, then hair color, like that. All characters, there is a reason behind all this character. A gene is controlling all this character. Each and every character is the expression of a gene, or a particular gene is expressing all this character. So you know that in a diploid organism, they have 2 copies of each gene, right? In a diploid organism, in the case of humans itself, you know, from each parent you are getting 1 set of chromosomes, right? So you have 2 copies or 2 alleles of a gene you have, right? So we call these 2 copies as a pair of alleles. So in homozygous condition, these alleles, this will be similar, right? Homozygous in the case just now we told capital T, capital T. This is the homozygous condition. Homozygous means both alleles will be similar. There won't be any difference between alleles. So if both alleles are similar, definitely their expression also will be similar, right? But in some cases like there is a heterozygous condition. If there is a heterozygous condition like this, capital T, small t, both alleles are different. So both alleles express in different way. So you know, in heterozygous capital T, small t, both alleles are different, right? So one of them may be different, or their expression may vary slightly. Or why it is different? Why this allele is different? Due to some kind of changes that has undergone. Okay, so there will be some kind of modification in the informations also, right? If this in homozygous condition, capital T, for example, we are saying this plant height in capital T, both informations are same. So the expression also will be in a similar way. But here, when it comes to heterozygous condition, here you can see in heterozygous condition, both alleles are different. Why? What is the reason behind that difference? The changes or the modification that has undergone for some modifications. That's why it is forming different alleles, right? So these difference in alleles, that definitely will be there in their expression also, right? So there will be some information changes. If this allele is having some kind of information or code, gene code will be there. This allele code will be different, and there will be slight difference in this allele code. Genetic code also, right? So there will be some kind of modification in the information that a particular allele makes. Okay. So every gene that contains information to express a particular trait. So in a diploid organism, there are 2 copies of each gene. We have already told these copies of gene or different forms of same gene. They are called as alleles. So these 2 alleles, they won't be always identical, right? In the case of heterozygotes, both alleles are different. But in homozygous condition, both alleles will be similar. In heterozygous condition, these alleles will be different. So one of them may be different due to some changes in that. Okay. Due to some changes in that allele will be different, and this difference, allelic difference, makes difference in their informations also, or that modifies the information that a particular allele is having. Okay, so the same difference in allelic difference that continues in the informational difference also. One allele will give one particular information, and the recessive allele here, one dominant allele will give one information, and the recessive allele will produce the information which is having some variations or difference. Okay, explaining all these things because we need to get an idea about the role of a gene, or what a gene does in our body. Okay, how these characters are expressed. That's why I'm explaining all these things. So in the next chapter, we will be learning about gene expression, how a gene is getting expressed, or what is the role of a gene, or how a character is getting expressed. So I'll just give an outline now. Gene. You know that gene codes for, this codes for DNA. Then from DNA, what comes? RNA. DNA, from DNA, RNA is formed. Then this RNA codes for, this RNA that codes for protein. This DNA, RNA and protein, this is called as central. DNA, RNA is forming, and from RNA, protein. This is called the central dogma. Okay, central dogma of molecular biology. That is the basis for all this gene expression or character expression. Okay, so gene codes for DNA, and this DNA, from DNA, RNA is forming, and RNA along with ribosome that forms the protein. And this protein gives different expressions or genetic expressions in our body. Okay. Here, a gene is having a particular information. For what? For producing an enzyme. Okay. With the enzymatic production that starts from a gene. So a gene is having different kind of information in that DNA, and that DNA is producing RNA, and that RNA decoding of that RNA, that forms the enzyme. Okay. So here the information for producing an enzyme that starts from the gene. Okay. So that gene is having, that particular gene, that's what we are talking about. That particular gene is having two copies or two alleles, right? A particular gene that is having alleles, two copies. These are called as alleles. So different chances are there for the expression, for this allelic expression, right? Here a gene is having two different forms. These are called as alleles, right? So there are different chances, or there should be some kind of difference in the expression of these alleles, right? So in normal case, these alleles codes for a normal enzyme. How it is forming from gene, and how it is starting, right? A gene is having enzyme of gene. Okay, enzyme a result of gene expression, right? So normal case, these alleles, both alleles code for normal enzyme, right? Usually what an enzyme does, this enzyme will act with substrate, right? substrate and that forms a product. Human nature act with substrate to form product. So that's how a chemical reaction is taking place in our body, right? So normally an allele produces normal enzyme, right? Normally allele produces normal enzyme which is needed for the transformation of a substrate into product, right? So we can say that the chances of this modified allele, a normal allele will form a enzyme and this enzyme will react with substrate and it will form a normal product. That is the normal or regular process, right? So if there is any change in that allele or any modification in that allele, any changes happens to that normal allele, that codes for enzyme which is needed for substrate transformation, right? So if any changes happens to that normal allele, that codes for the enzyme which is needed for substrate transformation. That changes in allele may cause the production of normal enzyme. Okay. How it is affecting? See, normal case, enzyme. An enzyme is forming and from a gene or from an allele, an enzyme is forming, right? This enzyme will be normal because here normal allelic expression. There is no change. That is a good allele. So it is forming a normal enzyme and that normal enzyme is acting with substrate and it is forming a normal product, right? But here by chance any modifications are happening in this allele, that there will be a change in the enzyme and again when it is reacting with substrate, the product, what we can imagine, that may affect the production of normal enzyme. But here theoretically that modified allele could be responsible for the production of the normal enzyme or less efficient enzyme or a non-functional enzyme, or there won't be no enzyme at all. All these chances are there, right? If there is any change in that allele, that may result in different cases, right? I'll explain you how it is. Here, in normal case, what is there? A gene forms enzyme, right? A gene or a normal allele we will write. Normal allele, okay? This forms the enzyme, right? And this enzyme is needed for the substrate, right? Substrate. So if there is any changes, when the allele, any change in allele, right? If any change, this normal allele is getting changed, there are 3 chances, correct? There are 3 chances. In first chance, this gene or the allele, here any changes in allele, there are 3 chances. First one, there will be a change in allele, but it will produce enzyme, right? But this enzyme will be, there will be a normal enzyme. Sometimes it may be a normal enzyme or there is a chance of less efficiency. Sometimes it may result in normal enzyme, but in some cases it may be that normal enzyme, but it is not efficient. It may not work in proper form or proper way. It may not be efficient to produce a proper product. Okay, when it is acting with the substrate, it may not result in a proper product. That is the first case. And some changes in that allele that may result here. If that allele is changing, change in allele, that may result in enzyme formation. In this case also, enzyme formation may happen, but here that may be non-functional enzyme. Okay, that enzyme may not be functional, it may not work. Here that enzyme is formed, it will work, but not efficiently. It may not form product in a proper way. That do not show that complete efficiency of that enzyme. But next case, when changes in this is the first case, right? In second case, changes in allele means it may produce enzyme. Enzyme will be there, but it will not work or it may not be functional. Okay, this is the second chance. Next, third chance is this gene, this changed gene or changed allele. If there is any change in allele or gene, that may not produce any enzyme. No enzyme. Okay? No enzyme is produced. This is the third chance. So all these things we can expect, right? If there is any change, normally a gene or the alleles, normal alleles will code for the enzyme, and that is needed for the substrate for acting, and then after an enzyme and substrate action only a product will be formed, right? Enzyme substrate, after that a product formation is taking place. But if there is any change in that allele or any difference in that allele or any problem in that allelic changes, that may result. Here in first case, enzyme will be formed, but that will have very less efficiency. It may not be efficient. In the second case, the changes in allele that forms enzyme, but that enzyme may not be functional. That may be non-functional. Then in the third case, your gene or allele is changed or any difference in that gene or allele, no enzyme is produced. So we can expect the allelic changes in allelic changes, we can expect all these three conditions. Okay. So next comes the concept of codominance that we can explain in next class. So do you have any? So this is the condition. From this we are going to next step. Okay. So you should be clear with this gene, how it is creating and how it is expressing, or what is the concept of this dominance and what a gene does actually. So you should be clear with all these. Do you have any doubt on this? Okay, hope up to this everything is clear. Deepa, do you have any doubt? Is it clear or any confusion up to this? Is it clear? Okay. Okay, so next codominance we'll explain in next class. Thank you.
Thank you, ma'am.